Wanted to thank the conference organizers. Very nice to see this whole community interested in autism. And I've enjoyed the presentations today. And want to talk to you today about our findings. A little bit higher. Okay. That better? Would like to talk to you this afternoon about our work, about a somewhat out-of-the-box approach. Can you hear back there? Okay. To a specific neurodevelopmental disorder that we think may have applications more broadly. I'm going to start with the index case. I'm a professor at the Technion. I have a history of working my laboratory devoted to functional genomics. For personal reasons, moved my lab over a couple of years ago to study this particular case and to try to find translational interventions that could ease the load in this particular case. We start with a child at the bedside at age 4 with severe ID. He was averbal and limited social interactions. Drug-resistant epilepsy, up to 25 tonic-clonic seizures a day, and many more smaller seizures. Exomic sequencing of the child identified a de novo missense mutation, a valine for alanine at residue 350 in the X-linked IQ SEC2 gene. Evidence that this was the smoking gun came from other children with similar mutations at the same residue, albeit a different amino acid substitution. And this was predicted to interfere with some of the functionality of the protein, in particular the binding of calmodulin to the particular domain in which this mutation was found. Motivating our research from its initiation was an interesting insight that we obtained from the bedside in this child. And that was the recurrent benefit that he had from fever. From infancy, whenever he would have a fever of 38 degrees or greater, he would have complete cessation of his seizures, and he would, after the fever had abated, a 2 to 3 week window of which he did not have seizures. Also during this period, his behavioral interactions were significantly improved. He remained averbal, but he interacted in a more positive way. So what I'm going to talk about today is what we've learned about the mechanism for this benefit of fever, at least on epilepsy, and we'll move on to other parts of the syndrome. And will a similar benefit occur if we purposefully induce fever? Okay. The history of fever through the ages, starting from antiquity. Hippocrates, I saw in a previous lecture this morning, referred to. Rava, one of the Tanaim, Rufus, student of Hippocrates, all found that fever, in fact, inducing fever, had benefit on a number of disorders, including epilepsy. With time, perhaps the Middle Ages and the advent of the plague, seeing that fever was a sign and a marker of serious disease and death, minimized enthusiasm for fever, and of course being recognized as a cause of disease. Currently, many pediatricians, many will treat fever at all costs, itself viewed as being an actual disease in and of itself, which of course is erroneous. The association of fever with clinical benefit has been demonstrated in several different arenas, particularly relevant to this audience. With regard to autism, first demonstrated at the Kennedy Institute in Hopkins, when an epidemic swept through the institute, this is in 2009 in this paper in Pediatrics, for a marked improvement in the behavioral interactions, in the robustity, the ability to speak in a large number of patients at this particular institute. This has been replicated in multiple other cohorts. That a significant percentage of children with autism with fever will start to have improved social interactions. The Nobel Prize in 1927 was awarded for the induction of fever to treat a particular type of dementia. Alzheimer's disease has been talked about as another disorder which may be helped by fever based on some longitudinal studies in Finland. Part of the problem with this area of research and this particular application to treat disease has been the lack of mechanistic studies as to why fever might be beneficial, and also hampered by the lack of appropriate animal models in which these mechanisms may be investigated and elucidated. So how might fever protect from disease? From bacteria to man, we have a cellular response to fever, known as the heat shock response, whose major function is to produce chaperones, which bind and help refold abnormally folded proteins or mediate their elimination. So we sought at this point, at which point at this time there were 4 known cases in the world, to develop tools to understand how this mutation mediate disease, true personalized medicine approach, I believe, and how fever may indeed rescue this mutation. It was known previously that this gene, the product of this gene, is expressed more prominently in the brain, in particular in the hippocampus, found at the postsynaptic density of both excitatory as well as inhibitory interneurons in the hippocampus, as I said, at the PSD, and associated with the NMDA receptor. It was known the function of this protein is to act as a guanine exchange factor, or GEF, mediating the exchange of ADP for ATP, particularly on ARF6. ARF6 main function is to mediate the trafficking of numerous neurotransmitter receptors, notably the AMPA receptors, as well as to have been demonstrated to play an important role in dendritic spine maturation. Poised, therefore, to play a major role in intellectual development as well as in other disorders such as autism. The mutation in the residue 350 was in what's referred to as the IQ-like domain. This domain of the IQ protein mediates the binding of calmodulin, which, as I will show in a few moments, mediates the allosteric modulation or modification of the SEC7 catalytic GEF activity of IQ-SEC2. So this mutation was predicted to interfere with the interaction of calmodulin and thereby to interfere with its allosteric function of its SEC7 activity. We initially approached this using a very generalized approach to look at protein-protein interactions called Lumiere, in which case you tag 2 proteins looking for try to quantify their interaction, in this case tagging calmodulin with FLAG, tagging IQ-SEC2 with luciferase, and we're able to quantitate the degree of interaction in cells that have been transfected with these different constructs, either stably or transiently. We found a dramatic reduction, as predicted, in the interaction of IQ-SEC2 mutation of A350V with calmodulin. The significance of this lack of binding to calmodulin comes from studies on trying to understand what the function of the wild type is. Basically, as I showed in the previous slide, there's a SEC7 domain responsible for the GEF activity of IQ-SEC2. This involves an interaction, a direct interaction between the SEC7 domain and ARF6. Normally, when calmodulin is bound to IQ-SEC2, it interferes through a specific HIN region at residue 342 in the interaction between SEC7 and ARF6. Calcium influx in response to neurotransmitter results in the disassociation of calmodulin from the IQ, and thereby allowing the SEC7 ARF6 interaction. Mutations such as A350V, which interfere with calmodulin binding, thereby constitutively allow the ARF6 SEC7 interaction, and would be predicted to result in much higher levels or constitutive elevation of ARF6, which is normally seen in the wild type situation only with calcium transients. And in fact, that's what we've published and demonstrated with this particular construct. It is a constitutive upregulation of ARF6 with this particular mutation. This led us to propose the following model based on what was known about the wild type, based on the predicted elevation in ARF6 activity and the known ability of ARF6 to mediate the downregulation by receptor-mediated cytosis of AMPA receptors, in particular GLU-A2. This is work that had been done by Josh Brown and Nishat Gurgis some years previously, showing that this is the normal function of ARF6 activation. So we predicted that when you overactivated ARF6, you would in fact see a downregulation of AMPA receptors. In order to study the biology of this mutation and IQ in general, it was necessary to develop molecular tools that would allow us to study the pathophysiology of this disorder as well as the potential benefit from fever. And as is customary today in personalized medicine, the two most prominent tools that we developed was one, a CRISPR-generated mouse. The mouse and human gene are 99.7% homologous at the amino acid level. So the amino acid we could mutate in the mouse that was identical to the human disorder. And so we were able to generate such a mouse. These mice, as we'll show, faithfully recapitulate in mice several of the features of the IQ phenotype. We also took cells from the child with the mutation and made induced pluripotent stem cells. And in collaboration with Shani Stern from Haifa University, these were differentiated into hippocampal dendrite cells. And the biology of these cells, the electrophysiology of these cells was studied, and I'll be showing some of that data. All this data now has been published, both with regard to our mouse model as well as our iPSC model. So coming back to this prediction, this is, of course, initially just in HEX cells. We would predict that this mutation would result in a downregulation of surface AMPA receptors on excitatory and inhibitory neurons. And we've been able to demonstrate this in a variety of models, which I'll run through here. First, with regard to in the murine hippocampus, using 3 different complementary approaches, we were able to show there's a significant downregulation, specifically in GLUA2 in the hippocampus of our A350 mice. Using the induced pluripotent stem cells that are differentiated into hippocampal neurons, we also corrected the mutation in the child's cells using CRISPR, so we had an appropriate isogenic control in which to look at the pathophysiology, in this case, AMPA receptor density, and the cells are differentiated over a period of literally weeks. When the cells become mature and form networks, we saw a significant downregulation in the amount of surface AMPA receptors, similar to what we saw in the mouse model. Also in embryonic neuron cells, not only in neonatal cells, we saw a similar phenomenon. So this reduced AMPA activity would be predicted to have impact on the electrophysiological properties of the neurons derived from these mice, as well as to have an effect on synaptic transmission. I want to show you data now that in fact reinforces that hypothesis. This is first with hippocampal slices. This is done in collaboration with Nat Jurgis from University of Wisconsin, in which we're looking at the stimulus response in CA3, looking at CA1 activity. Seeing it in orange is the mutant slices, as opposed to blue is the wild-type slices. A significant reduction in synaptic transmission in the A350 slices in response to induced stimulation. This is in a mature network of A350 dendrite cells from the, as I said, from the iPSCs, comparing the control in blue, in other words, the CRISPR-corrected isogenic control, compared to the mutant shown in red. A dramatic reduction in spontaneous synaptic transmission. These are iPSCs that we're looking at here that are dramatically reduced in this relatively mature network of neurons from the mutant cells. This is now looking at the neurons derived from neonatal mice at 1 day, looking at the frequency, in this case, specifically at AMPA-mediated mini EPSCs, small exercitatory postsynaptic potentials. In this case, we can particularly say these are due to changes in AMPA because we're able to block other types of receptor-mediated transmission through NMDA or GABA and the presence of these inhibitors, and showed that you see at the top of this slide, looking at the mutant, a dramatic reduction again in the amount of mini EPSCs in the mutant as compared to the wild type. Both did not reach significance in the amplitude, but a more dramatic reduction in the frequency of these mini EPSCs in the mutant. Moving on from the cellular model now to the animal model with regard to characterization of the phenotype that we can then use as a tool to probe mechanisms. We've looked at a variety, and much of this work has been done in collaboration with Professor Shlomo Wagner at University of Haifa. We've been able to look at seizures. This is looking at impaired learning paradigms, such as the water maze, and impaired social interactions. Draw your attention particularly to the social interactions. Again, this was done with collaboration with Professor Wagner. Two paradigms that were particularly powerful. One was looking at the interaction between a male affected with the mutation and his preference for a male versus female mice in a variation of the three-chamber arena. And the normal mice, of course, refers to associated with the female mice. These particular mutation resulted in an absence of preference for the male versus the female mice. The most robust phenotype, however, we saw was in the amount of ultrasonic vocalizations shown in the right-hand part of the slide, that the normal mice, when a male mice associates with in a freelance open field, has a dramatic induction of ultrasonic vocalizations in part of his courting mating protocol. The mutant mice, however, are by and large mostly silent and do not engage in such and demonstrate such vocalizations. So these are some of the models that we've gone on to use to investigate therapies that I'll be touching on now. But I didn't mention the seizures. We also characterize seizures in these mice, both in the female and as well as the male. The female heterozygote has about half the frequency as we predicted from exon activation as in the male. Approximately 50% of the mice have seizures, shown in the Kaplan-Meier here, between days 15 and 20, they go into status and die. Some mice do recover, but approximately 50% of the mice die from seizures, specifically between days 15 and 20. After day 20, there is no more seizures. We do see sharps after that, and there are small spikes, but there's no seizures after day 20. The mice have died that have had seizures. I'll show you just some, we've been able to get EEGs in collaboration with Professor Eric March from CHOP, we've also captured these by video as well as by EEG, which is very challenging between days 15 and 20 due to the scalp of these mice, as well as looking at sharps in the adult mice and noting increased delta wave frequency in the adult mice as well. This is an example of seizures in these mice. The upper panel shows 14 such seizures that are associated by video with the dramatic posturing of the mice consistent with the seizure, and the bottom shows seizures in additional mice as well. So now with these tools at hand, we felt we were in a position to try to understand the mechanism by which fever could rescue, or did it rescue, these mice, similar to what we saw with the child. I'll be talking about our preclinical studies, looking in particular at the interaction between IQ and its binding property to calmodulin, GF activity of IQ, and the mouse models, as well as what we've done in the child himself. So first I want to talk about how do you generate a fever? You know, there's many different possibilities here. Classically, in antiquity, one of the most prominent ways was to actually inject pyrogens. And the Nobel Prize in 27 was done by injecting malaria, believe it or not. But we don't believe that that is necessary. And you know, fever therapy is actually used today in some paradigms. But the basic idea is that there is a hypothalamic set point. And either through endogenous pyrogens, such as interleukin-1, or exogenous pyrogens, such as endotoxin, this results in a resetting of the hypothalamic set point that results in a fever. Another way, though, to generate an elevation in body temperature is taking advantage of what we know about how body temperature is regulated. Of course, when we get overheated, we sweat and we vasodilate, and this is the mechanism by which we can maintain our temperature in a normal range. However, when you're put in a situation, probably that occurs here sometimes in the summer, that you're unable to sweat because the outside temperature exceeds 37 degrees or even higher, very rapidly you can raise the body core temperature because you're unable to dissipate the heat that is generated as normal part of metabolism. So these are two different mechanisms by which one could generate a rise in body temperature. We've monitored the body temperature, the core temperature, not the skin temperature, the core temperature of these mice using a mini transponder, which is ideal for mice of this age, which can be implanted percutaneously. and then we're able to monitor the temperature of the mice using remotely. We're also physiologically able to monitor heat shock in these mice and elevation in temperature, particularly in the brain, by monitoring the induction of specific heat shock proteins and their RNAs. So, as I said, the initial strategy we took was using pyrogenic substances. We did not find, however, in these relatively immature mice, day 14, this is when, again, seizures are occurring at this window, that they were able to have an induction of fever. This may be similar, many of you may be aware, that many children before 2 months of age, early in development, actually lack an ability to induce fever. And this has been seen in other animals as well. So we turned to the alternative mechanism to try to raise body temperature, and that was through thermal stress. We adopted initially a paradigm which had been used in military medicine, actually, to elevate the body temperature in a very controlled way. And this was by incubating the mice. Normally, of course, in most of our facilities, we house the mice at about 23 degrees. In this case, we house the mice at 37 degrees for the period of the seizure susceptibility. When we monitor the temperature of the mice gradually over a period of 48 hours, they develop an increase in body temperature to 30 to 38 degrees. They don't get any higher than 39 degrees over the entire 5 days. Shown in the bottom part, we can see an induction of HSP70 in the brains of these mice in response to this particular stimulus. And what we found was there was a dramatic reduction in mortality and seizures in these mice. Basically, we saw from approximately 50% incidence of mortality between days 15 and 20. This was reduced to about 2% in the mice with the thermal treatment. And they did not go on to have seizures later in development. We then asked, okay, so we've stopped seizures in these mice. What are the implications for later in development? In particular, this very robust finding we found with regard to ultrasonic vocalizations. And again, with collaboration with Professor Wagner, we looked at mice that had been treated as pups, what their behavior was now when they reached maturity between 2 to 3 months of age with regard to stimulated or appropriate induced vocalizations in response to a female stimulus. And the right-hand part of the slide, again, reminds you what we saw initially in the wild type versus the mutant. In response to this mice that have been treated in children as pups, excuse me, as with regard to fever, this fever induction, when they became adults, a very significant improvement in their particular stimulus-induced vocalization. Now, that's shown in, what is the significance of this? These increased contextually appropriate vocalizations in the adult mice treated with thermal stress as pups, we believe may be due to stopping the seizures and permitting a more normal trajectory. The implications, I think, are significant. If one could stop the seizures, it may prevent a more normal development. This is hardly surprising, and many of you are familiar with epileptic encephalopathies. So easing the burden of that is contributing to the genomic phenotype of this disorder. Does this have any translational relevance in man? Based on these findings, the child himself has undergone jacuzzi treatments, okay? He sits in a jacuzzi for 15 minutes, twice a day. Jacuzzi set, as is customary, to 40 degrees. We've been able to monitor his core temperature, getting up in this particular paradigm to about 38 degrees. We've seen a dramatic reduction in his seizures, both quantitatively monitoring his seizures, as well, and a dramatic improvement in his EEG in terms of the number of epileptiform spikes, an increase in wakefulness, his interactions. He's still averbal, but undergoing aggressive ABA. So, how might this be working? How might fever be providing benefit? Okay. Our working hypothesis has to do with HSF1. And this is based on the fact that we can by inhibiting HSF1, we're able to prevent the seizures. We've been studying in my laboratory several different mechanisms by which this may be mediated. Just for lack of time, I won't be able to go through these different mechanisms, but we're trying to elucidate what is the pathophysical mechanism by which fever provides this favorable response. This is one example that provides some evidence of the fact that we're actually refolding the abnormally folding IQ protein. This is done with looking at the IQ calmodulin interaction. This is an increase when the cells are incubated at 40 degrees. This is blocked by an inhibitor of HSF1 that can block this induction of the improvement interaction. With regard to the electrophysiological activity of the neurons, we're able to rescue the diminished mini EPSCs due to AMPA in these cells by the heat shock in the neuronidal cells, shown in the right-hand part of the slide, by this treatment. So, summing up now, the thermal therapy that we're proposing, in order to be practical, cannot be, of course, continuous fever therapy. We've been able now to demonstrate with simply a 15-minute treatment in these mice results in a dramatic reduction in their seizures. Working on ways to enhance and to model this heat shock treatment to make it something that can be translated more readily. I should add, this now has been moved. We've moved on to additional children that have been able to, one particularly in Greece, now using a similar treatment with a different mutation in IQ, which appears to be deriving significant benefit as well, similar to what we've seen here. We believe this can also be, there's other children as well with fever-induced improvement in their symptomatology with IQ mutations, and we're studying these mutations as well. We think this may have relevance for a variety of other diseases as well, which have similar pathophysiology. We talked this morning about networks, and that many of these genes overlap in their pathophysiology, and we're investigating the potential use of this therapy in other diseases as well. So I wanted to thank members of my laboratory and my collaborators, and thank you for your attention.
Thank you, everybody. This was really fascinating, and we have really time for one quick question, so please let them.
Thank you very much. Very fascinating talk. Thank you very much. There was a recent paper which also talked about fever and autism in a mouse model. And in their model, so they show that fever caused perturbation of IL-17.
That's correct.
Which they can then inhibit, so that inhibited the positive effects of the fever. I'm curious if you think there could be, or there's a reason to think that there wouldn't be any involvement of the immune system in your model, or you think those are two completely different things?
It's a very good question, and actually we've looked at IL-17 in this model. IL-17 is actually a heat-inducible gene. Okay, it is. We've seen actually there's an induction of IL-17 in the hippocampus of these mice. At baseline, there's actually a lower level of IL-17 in the mutant compared to the wild type. So it's very possible that IL-17 could play an important role in the pathophysiology that we're seeing here. Sure.
Okay, thank you so much. Unfortunately, we don't have the time for more questions.